आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज आपकी आवाज, विज्ञान की आवाज, की आवाज।
We bring you all a podcast series on STEM popularization, that is, science, technology, engineering, mathematics, and medicine, supported and catalyzed by the National Council for Science and Technology Communication, the Government of India. Program is produced by Media for Community Foundation.
Welcome back to the podcast from the Indo French Center for the Promotion of Advanced Research, also popularly called as CEFIPRA. We are dedicated to showcasing the groundbreaking collaborations between Indian and French scientists that are expanding the frontiers of knowledge. Now, let's understand more from our scientist.
Hello, I'm Kaustuv Sanyal. I'm a professor and chairman of Molecular Biology and Genetics Unit at Jawaharlal Nehru Center for Advanced Scientific Research in Bangalore, India. I have obtained my master's degree from Madurai Kamraj University, and then I did my PhD in East Genetics at Bose Institute in Kolkata, before I went for my postdoctoral work at the University of California in Santa Barbara. I joined here at JNCASR in 2005 as an assistant professor, and my research interests include molecular understanding of the process of chromosome segregation, and I use various pathogenic budding yeasts as my model. So I started working on several proteins that are required for segregation of chromosomes during mitosis, which is the process that helps one cell to divide to give rise to two daughter cells. During my postdoctoral studies, I got interested in an organism called Candida albicans that causes various diseases in humans, especially those who are immunocompromised. At that time, with Professor John Carbon at University of California, Santa Barbara, we identified an important chromosomal DNA sequence which is called the centromere. Centromere assembles several proteins that form the kinetochore, which is the binding site of spindle microtubules that facilitates the process of chromosome segregation. So once we identified the centromeres of Candida albicans, we realized that unlike most organisms where the DNA sequence is conserved across centromeres in a given organism, in this case each centromere sequence is unique and different. When I started my own lab in JNCASR here in Bangalore, we got interested in understanding the centromere evolution by studying several pathogenic budding yeasts which are related to Candida albicans. While doing so, we realized the centromeres are the most evolving DNA sequences in the genome. That led to an interesting understanding that whether the centromere DNA sequences are the driving forces of speciation. So I have started the pathogenic fungal biology research at the molecular level in India, with a major focus on the fungal centromere biology. Though you know that centromere is a chromosomal locus that serves as a binding platform for mitotic spindle. So during in the initial phase of my research in India, we identified and analyzed centromere sequences from many major pathogenic fungal lineages, and we realized that centromere DNA sequences are evolving quite rapidly. The centromere DNA sequences can be quite different from each other even in the same species. That helped me to realize that there must be factors other than the DNA sequence that determine where the centromere should be formed. To test this hypothesis, we deleted a native centromere in Candida albicans, and we found that a region next to the native centromere, which we deleted, formed a new centromere which we termed as neocentromere. This suggested that centromere identity is not determined by the DNA sequence alone, but the spatial location of the region of a chromosome in fact determines where the centromere should be formed. We have performed more research in understanding the 3D conformation of the chromosomes using techniques such as chromosome conformation capture to understand the spatiotemporal regulation of centromere identity. We perform various collaborative research from scientists across disciplines such as mathematicians, physicists, and chemists. So one of such collaborative ideas came when a scientist named Prof. Was visiting India to attend a meeting in Delhi. We were casually discussing about exploring a library of Candida albicans. While talking to Christoph, we discussed about possibility of us utilizing Candida albicans overexpression ORFOM library that he has created with another collaborator. We were interested in understanding how what are the determinants of chromosome segregation, and this library we thought would benefit us to screen for factors that may regulate chromosome stability. So he immediately agreed to collaborate on this project, and we started writing up a proposal. So we had several discussions over Skype, and we came up with a proposal and submitted to CEFIPRA. And we were lucky that the project was funded and we started our collaboration. So one of the advantages of having a CEFIPRA project is the exchange visit. So I could go and visit Pasteur Institute in Paris, where my partner Christophe works. The French PI lab was really well equipped with various high throughput techniques. I learned various facilities that they have, and I could also interact with many other faculty members of the unit. I could exchange my research ideas and learned a lot. In fact, Christophe also visited our lab twice, and interacted with my students and postdocs here, and it was a very enriching experience for both of us. Eventually, when the project got started and was moving well, my PhD student who was involved in the project had to learn how to do high throughput assays and high throughput screens. So she went to the Pasteur Institute in Paris to learn the techniques because we were not aware of such techniques, and she spent about 3 months there. Thanks to Cefipra again, that made it possible, and she came back and we developed the same systems and same facility right here in Bangalore. So by doing so, we screened about 1000 genes of Candida albicans, and we analyzed what is the effect of overexpression of each of these 1000 genes on chromosome stability, and finally identified one of the factors which he named as CSS6, that is present only in Candida albicans and some other related pathogenic yeasts. The gene, the new regulator of genome stability that we found in Candida albicans and named as CSS6, is an interesting protein, because what we realize that when we overexpress this protein, then the cells are arrested at metaphase stage of the cell cycle, and they cannot proceed to the next stage of the cell cycle. And when we depleted the protein, and we found that this protein is essential for viability of Candida albicans cells, because they were again arrested and in the cell cycle. But interestingly, unlike the metaphase arrest that we observed when we overexpressed the protein, when we depleted it, we found that the cells are arrested at the anaphase stage of the cell cycle, which means that these cells with depleted levels of CSS6 could not proceed to mitotic exit. So, in other words, what we have shown that we identified a unique protein that is present only in Candida albicans and related pathogenic yeasts and not present in humans, is so finely regulated that its abundance or its depletion can cause cell cycle arrest and thus death of Candida albicans. So this raises a possibility of targeting CSS6 as an antifungal drug target, which since it doesn't have a human homologue, so if we can identify a specific inhibitor of CSS6 function that can specifically stop cell division of Candida albicans without much effect on humans.
This episode is brought to you with the content support from Cefipra and the technical support from the Sparrows team. We extend our heartfelt thanks to the Director of Cefipra, Professor Nitin Seth and Dr. Payal, for their continuous guidance and support. Stay tuned for more episodes and thank you for joining.
आइए हम सुने विज्ञान की अनकही कहानियाँ, आइए हम सुने विज्ञान की अनसुनी कहानी।